idea of bringing all of us here for this absolutely fantastic opportunity. Thank you, Winstep Forward, and everyone, Eric, Lexi, Navida, who helped us so much. Okay, so now let's talk about our project. But before that, thank you to this absolutely beautiful group. Some of them are here too, who I really love, and I don't know how I'm going to say bye to them. Okay, so expecting that most of you are from different departments of maths and physics and don't know much about what we do, I have made this presentation very generalized, and I'm going to give you a broad picture of what I'm doing. And if you want to know more, definitely come stop by my poster, right? So my project is on transcription translation coupling. Most of you probably don't know what these two terms are. So let me tell you with this kitchen theory that I made. So let's think of our human body. We are made up of cells. Let's think of each cell to be a kitchen. What do we do in a kitchen? We make dishes. So let's think of the most important dish that we want to make in the cell, and that's proteins. They are very beneficial for us. And there are people who are doing it in the cells. We have got two chefs. One of them is RNA polymerase, and the other is ribosome. Okay? And how they do it is one of the most important theories in biology called the central dogma of life. So we have the DNA on top, and it gets produced into the mRNA through the process called transcription. So you already know one of the terms in my project. And the chef that does it is RNA polymerase. The mRNA then gets produced into what we want, the food, protein, through the process called translation. And who does that is ribosome. So this is what transcription and translation is. But what is coupling, right? So in prokaryotes and eukaryotes, there's a difference. In eukaryotes like us, transcription and translation occurs in two different compartments because we are complicated kitchens and we have compartments. But in case of prokaryotes like bacteria, who are simple cells, they all have it in the same place. And therefore, there is a possibility that both of them can be coupled. The expressome is the visualization of how these two things can be coupled. And I'm grateful to say that Dr. Robert Landing I'm working under is one of the person who coined the term. And this is more specific, the structure. So this is the ribosome on top, the second chef, and this is RNA polymerase, the first chef. And both of them kind of are together, as you can see. Now, think about it. How can coupling happen? So if there are two chefs and we can bring both of them together to work together, coupling can happen, right? So it's like bringing both the processes together. And how this happens is through NUSG. This green big protein is NUSG, which is a transcription factor, and it has two arms, the N-terminal domain and the C-terminal domain. And what it does is, using these two hands, it pulls both the chefs close to each other so that they can couple the two processes. Now what my project was, I was trying to focus on NUSG and see where all is it located on the DNA. For people who understand biology, I'm going to tell you quickly what I was trying to do. So NUS-E is a ribosomal protein to which NUSG binds through its C-terminal domain. And if I'm not wrong, and I'm sorry, and I'm going to epitope tag NUS-E and then chromatin immunoprecipitate it. So when I bring down NUS-E, what happens is that since NUS-E is bound to NUSG, it brings NUSG down as well, which is bound to the DNA. And then I sequence it to find out where NUSG is present on the DNA, which is by ChIP-seq. But the main question is, why are we doing it? The first thing that each and every scientist asks before doing anything is why. So as I've mentioned in red, we're trying to unfold the recipe of life, as I've been talking about, the central dogma of life. We know that we need proteins, and we know that DNA makes it, but we don't exactly know how is this process going on. We don't know exactly how these chefs work on transcription and translation and make proteins. And what happens when we know this recipe is that we can control the protein, which we want. So if we want the protein, we can make more of it. If we don't want the protein, we can make less of it, right? That's the power of what the Landing Lab is trying to do. trying to find out the mechanisms behind transcription and translation. So I have a lot of results as well, but you can talk to me. I would end it with these quotes that I've been collecting when in my visit here. Amazing scientists who told me so many things which are so inspiring. And the last thing that I want to tell all of you students here going to be scientists is that what is expected of all of us is to use this knowledge to help mankind, to help nature, and to give. Thank you.